Welcome back to the Deep Dive, where we plunge into complex information and pull out the most important insights, giving you a shortcut to being well-informed.
Glad to be here.
Today we're embarking on a really crucial deep dive in the very core of what makes us, the human brain.
It's an incredible journey we're on, scientifically speaking. We'll be exploring the latest in single-cell RNA sequencing.
Right.
And its really profound impact on our understanding of brain health and psychiatry. It's truly a frontier, It's reshaping how we think about these incredibly complex conditions.
Moving beyond those broad categories.
Exactly. We're starting to look at the brain at its most fundamental level, cell by cell.
That's right. So our mission in this deep dive is to unpack what single-cell studies have revealed about neuropsychiatric disorders so far. We want to give you a comprehensive but, digestible overview.
And then look ahead, right?
And then turn our gaze to what researchers should tackle next. We're aiming to shed light on how these cutting-edge technologies are truly transforming our ability to trace psychiatric disease.
Yeah.
Well, gene by gene, cell by cell.
It really is like having a high-resolution microscope focused directly on the brain's internal machinery, something we couldn't do before.
Okay. So, to set the stage for this detailed exploration, let's begin with a crucial question. What do we currently know?
Let's start there. We'll start by looking at the wide array of disorders that are already under the single-cell microscope. It reveals the immense scope of this new scientific approach. As segment 1, what disorders have been studied?
Right, so when we talk about single-cell RNA sequencing, it's not just a niche technique for one specific illness, is it?
Not at all. What we're seeing across the scientific literature is that single-cell and single-nucleus RNA sequencing.
Right.
It's been applied to an astonishingly wide spectrum of psychiatric and neurological disorders.
It's like researchers are using this new lens to re-examine almost every major brain condition.
Precisely. On the psychiatric side, this includes the major players that affect millions globally. Conditions like schizophrenia, major depressive disorder, bipolar disorder, autism spectrum disorder.
The big ones.
The big ones, yeah. But the scope isn't limited there. More recently, these incredibly precise single-cell approaches have expanded to conditions like post-traumatic stress disorder or PTSD, epilepsy, traumatic brain injury, often referred to as TBI, and even neurodevelopmental conditions like ADHD and obsessive-compulsive disorder or OCD.
Wow.
It truly underscores the versatility and the diagnostic power of these techniques. We're getting insights at a resolution we could only really dream of a decade ago.
That's an impressive list.
Yeah.
And I understand that among all these, schizophrenia remains the most extensively profiled disorder when it comes to single-cell research.
That's correct.
It's almost like a prime focus for this kind of high-resolution scientific investigation. Why do you think schizophrenia has garnered so much attention with these new methods?
Well, schizophrenia's complexity, its severe symptomatology, and its notoriously challenging genetic architecture have made it a persistent puzzle for decades.
Right, it's been tough to crack.
Exactly. So this makes it an ideal, almost necessary candidate for the unparalleled resolution offered by single-cell analysis. Researchers can now dissect the disease at a cellular level, revealing insights previously obscured.
So what are the key areas they're focusing on within schizophrenia?
The studies strongly focus on several key areas. First, they're digging deep into the genetic risk, exploring not just single genes, but also polygenic theories.
That's the idea that many small genetic variations add up, right?
Precisely, that they collectively contribute to risk. They're also looking at common alleles and specific high-impact risk genes. For instance, studies are now using polygenic risk scores, or PRS.
Okay, PRS. How do those work?
Think of PRS as a way to sum up all the tiny genetic predispositions someone might have, painting a fuller picture of their overall genetic vulnerability.
A cumulative score.
Yeah, and these scores show higher readings in individuals with schizophrenia compared to healthy controls. And crucially, they're directly linking these cumulative genetic factors to observable brain structural changes.
So it's drilling down into not just individual genetic influences, but the cumulative effect of many, and then looking at the brain's internal systems. What other areas are they focusing on?
Second, there is a deep dive into immune dysregulation, exploring altered immune responses and inflammation pathways.
The brain's immune system.
Yeah, we're increasingly recognizing that the brain's immune system, particularly cells like microglia, plays a critical role in the development and progression of many psychiatric disorders, not just traditional neurological ones.
Okay, that makes sense. And the third area?
And third, researchers are intensely examining synaptic dysfunction. These are disruptions in the neural communication at the synapse level, that tiny gap where neurons talk to each other.
The fundamental communication.
Absolutely. This communication is fundamental to everything the brain does, from thought to emotion, so understanding how it breaks down is crucial.
So it's a holistic view, the genetic blueprint, the brain's immune system, and how neurons communicate. That really makes sense. Are there any specific genetic models or studies that have given us particularly powerful insights into schizophrenia, perhaps revealing direct causal links at the cellular level?
Absolutely. Take the 22q11.2 deletion syndrome, for example.
Okay.
This isn't just one gene. It's a significant genetic variant where a small piece of chromosome 22 is missing, encompassing multiple genes. It serves as a profound model for schizophrenia, but also for intellectual disability and epilepsy.
So shared pathways, maybe?
It highlights shared underlying biological pathways, yeah. Studies on patient-derived organoids, which are essentially miniature, simplified versions of human organs grown in a lab dish.
Right, the mini brains.
Kind of. With this deletion, they reveal profound impacts on neurodevelopment. They show lower proliferation rates and slower differentiation into mature neurons in patient neural progenitor cells.
Those are the early building blocks.
Exactly, the early building blocks of brain cells. And this is accompanied by reduced neurite complexity, meaning the branches that neurons use to connect are less elaborate and impaired maturation of excitatory neurons, which are crucial for transmitting signals.
And it affects other cells, too.
It does. What's more, even in supporting brain cells like astrocytes from these patient organoids, there are dysregulated genes related to neurogenesis, the birth of new neurons, and mitochondrial ATP synthesis, which is how cells generate energy.
Wow. So it suggests a really broad, cascading cellular pathology.
It really does. A single genetic deletion can have these profound and widespread effects across different cell types and developmental processes. It almost sets the stage for the disorder very early on.
That's really illuminating because it fundamentally changes how we view genetic risk. It's not just one gene causing one problem.
No.
It's about a small genetic change setting off a chain reaction that impacts the very blueprint of the brain. What about other genetic variants that have been studied?
Another major risk variant is the 15q13.3 microdeletion. Similar to 22q11.2, this deletion is strongly associated with schizophrenia, but also with epilepsy, ADHD, and autism spectrum disorder.
Again, that overlap.
Providing key insights into shared genetic vulnerabilities across these conditions, yes. Research shows this deletion specifically affects the development of interneurons.
The traffic cops of the brain.
Good analogy. Those crucial brain cells that help balance signals. It affects them particularly in specialized nurseries within the developing brain called the medial and caudal ganglionic eminences, MGE and CGE.
Okay.
This impacts how these early brain cells multiply and mature at different developmental stages. The gene KLF13 seems to play a major role here, and its absence can lead to marked behavioral impairments reminiscent of these disorders.
And there's a link to environmental factors, too.
What's particularly fascinating is that this differential impact on interneuron subtypes is also seen in models of maternal immune activation. It suggests a common etiology or origin for both genetic and environmental insults in brain development.
That's a powerful connection, linking specific genetic variants to broad behavioral changes and even to environmental factors. It seems like these studies are revealing the brain's weak points. Are there studies on individual genes that highlight specific cellular dysfunctions beyond these large deletions?
Yes, absolutely. Studies on SET1A insufficiency are a great example of drilling down to individual gene function.
SET1A.
This gene demonstrates a crucial role in synaptic vesicle exocytosis dysfunction, specifically in excitatory neurons.
Okay, unpack that a bit. Synaptic vesicle exocytosis dysfunction.
Right, it's a mouthful. Imagine tiny packages inside a neuron called synaptic vesicles filled with neurotransmitters, the chemical messenger.
Okay.
Exocytosis is the process where these packages fuse with the cell membrane and release their contents into the synapse for another neuron to pick up. So dysfunction here means the cell can't effectively release its chemical signals.
Got it. Communication breakdown at the release point.
Exactly. Furthermore, research links SED1A to schizophrenia-associated genes identified in human GWAS studies, which are large-scale genetic surveys. Genes like TEC2, SCAF1, and NENSUN6 are among those correlated.
So connecting the single gene function back to broader genetic risk.
Precisely. This work uses techniques like gene ontology and ingenuity pathway analysis to show that in excitatory neurons, the differentially expressed genes are enriched in terms related to sodium ion export, regulation of calcium, ion-dependent exocytosis, synaptic vesicle transport, and neurotransmission. It points directly to issues with how neurons manage electrical signals and release their messages.
So it's really getting down to the precise machinery of how brain cells communicate.
Yeah.
Okay, what about something like traumatic brain injury? How does a physical event like that fit into the picture of schizophrenia-like behaviors from a single-cell perspective?
That's another incredibly intriguing area, demonstrating the broad applicability of these techniques. Research on traumatic brain injury, or TBI, often what we call a concussion or more severe head injury,
Right.
has identified the increased activity, or upregulation, of a gene called secretogranin Tess, SCG2, in the brain's outer lobe, the cortex, after injury.
Okay, SCG2.
And this upregulation is directly linked to blood-brain barrier dysfunction, meaning the protective barrier between the bloodstream and the brain becomes leaky, and the emergence of schizophrenia-like behaviors.
Wow. So a physical injury can trigger these molecular changes.
It provides a molecular mechanism by which physical trauma can contribute to psychiatric symptoms. It highlights neuroinflammation and the infiltration of immune cells from outside the brain as fundamental biological dysfunctions that can trigger these complex conditions.
Fascinating. So the brain's immune system and its protective barriers play a role in trauma-induced psychiatric symptoms. Okay, moving on from schizophrenia, let's look at autism spectrum disorder, or ASD. How are single-cell approaches shedding light on this multifaceted condition, which also has a strong neurodevelopmental component?
In ASD, the research aims are quite broad, seeking to unravel its profound complexity. They include exploring the genetic architecture, the underlying genetic blueprint, elucidating mechanistic insights into specific genetic mutations, and examining gene-environment interactions.
And epigenetics comes in here too.
Especially through epigenetic mechanisms like DNA methylation and histone modifications. Epigenetics essentially describes how gene expression can be turned on or off without changing the underlying DNA sequence itself, often influenced by environmental factors.
Right, the switches.
Exactly. It's a comprehensive approach to understanding how both nature and nurture contribute to ASD.
And what specific genetic mutations are standing out in ASD research when looking at the single-cell level?
One key area involves SHANK3 mutations. Studies using SHANK3 knockout mouse models, where the gene is essentially silenced link abnormalities in quiescent neural stem cells to neurodevelopmental diseases like autism.
Quiescent stem cells. The dormant reserves.
That's right, ready to activate when needed. The SHANK3 mutation causes them to be overactivated, leading to problems down the line.
Is there any hope for intervention there?
What's even more promising is the exploration of how epigenetic modulation tweaking those gene expression switches might alleviate synaptic dysfunction and behavioral abnormalities.
How might that work?
Well, the speculation is that this could involve epigenetically normal newborn neurons contributing to proper neuronal maturation and integration, or maybe even secreted factors from the recovered neural stem cell population helping to restore synaptic functions. It's a potential pathway for intervention, which is exciting.
So not just identifying the problem, but also exploring potential solutions at the cellular and epigenetic level. That is really exciting. Are there other genes being investigated in ASD with these techniques?
Absolutely. CHD8 haploinsufficiency is another significant area.
CHD8.
This gene's impact is investigated through CRISPR-edited human cerebral organoids. CRISPR, the gene editing tool,
Right.
allows scientists to precisely modify DNA. This research demonstrates CHD8's profound impact on early neural differentiation, how early brain cells specialize and cell-type specific gene expression.
What does it affect specifically?
While studies on CHD8 show some variations, they consistently point to its crucial role in regulating neural development and its effects on cell signaling, including vital communication networks like CNTN and NRXN.
Okay. Any others?
Then there's RFX3 deficiency, which is also strongly linked to ASD. Studies show its critical role as a ciliogenic transcription factor.
Meaning it helps build cilia, those tiny hair-like structures.
Exactly. And its impact on neurodevelopment, glutamatergic signaling, and synaptic function in human neurons is significant. It directly influences neurogenesis and the expression of genes involved in synapse assembly and organization fundamental to brain wiring.
It sounds like a lot of these studies are looking at the fundamental processes of brain development and how even subtle genetic changes can derail them. Is there an immune component in ASD too, similar to what we heard about in schizophrenia?
There is, and it's quite significant. Recent findings indicate the activation of the IL-6HIF1A immune axis in microglia as a contributor to ASD pathogenesis.
Microglia, again, the brain's immune cells.
Yes, acting like tiny guardians. This activation suggests a specific inflammatory pathway. Single-cell sequencing has been instrumental in identifying potential diagnostic markers like CDKN1A, ETS2, LYN, and SLC16A3, particularly in what are termed reactive microglia, or MG2 cells, in individuals with ASD.
So specific microglia types are involved.
It seems so. The upregulation of JUN and HIF1A in these microglia highlights a specific immune response that contributes to the disease, making them potential targets for intervention or biomarkers for diagnosis.
That brings us to major depressive disorder, or MDD, and its strong links to chronic stress. How are single-cell studies unraveling the complexities there, especially regarding the brain's immune system?
In MDD, especially in the context of chronic stress, studies in mouse models reveal really significant alterations in microglia.
What kind of alterations?
These changes include noticeable morphological shifts like hyperramification, where the microglia develop excessive branch-like extensions.
So they look different.
They look different, and this isn't just cosmetic, it reflects an altered functional state. Alongside this, there's a robust downregulation, a decrease in activity of interferon-related genes, or IRGs.
Interferon genes?
Yeah.
Usually involved in immune response.
Yes, and this downregulation is particularly seen in a distinct subpopulation of what researchers are calling interferon microglia. It suggests a very specific and frankly surprising immune response to prolonged stress, where certain immune functions are suppressed.
Suppressed. That is surprising. Are specific genes or pathways identified in this microglial response, giving us clues about how this suppression happens?
Yes. The transcription factor ATF3 is identified as a negative regulator of IRGs in stressed microglia.
ATF3.
And it suggests an IFN-independent mechanism for immune suppression. This means the suppression isn't happening through the usual interferon signaling pathway, which is quite a novel finding.
How strong is the evidence for ATF3?
What's compelling is that there's a significant overlap between the genes downregulated in stressed microglia and those upregulated in ATF3 knockout macrophages. This strengthens the hypothesis that ATF3 directly contributes to this repression.
Interesting. And you mentioned a link to physiology.
Additionally, yes. In conditions like temporal lobe epilepsy, or TLE, which often has co-occurring depression, decreased heart rate variability, or HRV, parameters correlate with depression severity.
HRV, the variation in time between heartbeats.
Exactly. This is crucial because it links central nervous system activity, especially in brain areas like the nucleus tractus solitarius, to peripheral physiology, the body's more widespread systems. It highlights that intricate brain-body connection in these complex disorders.
Speaking of epilepsy, how are single-cell approaches detailing its psychiatric connections, particularly with depression?
Well, in TLE, studies reveal that the severity of depression-like behaviors in mice is correlated with both seizure frequency and alterations in HRV, reinforcing that brain-body link we just mentioned.
And at the cellular level?
At the cellular and pathway level, we see elevated mitochondrial-related gene expression in glial cells and enrichment of the GABAergic synapse pathway.
GABA, the inhibitory neurotransmitter.
Right, so a disrupted GABAergic synapse pathway implies an imbalance in the brain's excitation and inhibition, which is crucial for normal function.
And specific neuron changes.
What's particularly striking is the specific upregulation of inhibitory neurons in the nucleus tractus solitarius. NTS, that brainstem region critical for regulating autonomic functions in TLE mice with depression.
The NTS again.
Yes, alongside an enriched long-term depression pathway in these neurons. This points to very precise cellular and circuit changes underlying the comorbidity of epilepsy and depression, suggesting shared cellular vulnerabilities. Segment 2, what brain regions and cell types are involved.
Okay, so we've covered the broad spectrum of disorders being investigated with single-cell sequencing. Now let's zoom in even further. Where exactly in the brain are these changes being observed? And which specific cell types are taking center stage in this groundbreaking research?
Okay. The prefrontal cortex, or PFC, is consistently identified as the primary hotspot for single-cell research in neuropsychiatric disorders.
The brain's executive control center.
Exactly. Responsible for planning, decision-making, personality. Specific Brodmann areas, BAs, within the PFC, like BA9 and BA24, are frequently studied.
Are other areas implicated too, particularly in schizophrenia?
Yes. Beyond these, research in schizophrenia also highlights decreased gray matter volume, meaning less brain tissue, in other key regions. These include BA13, part of the insula.
Involved in emotion and self-awareness.
That's right. Also BA23, BA24, and BA31 within the cingulate cortex, important for emotion and memory, as well as BA25 in the ventromedial frontal cortex, implicated in mood regulation.
So it seems specific structural issues in these areas might link to symptoms.
It suggests that, yes, this pattern suggests that certain structural abnormalities in these specific Brodmann areas may be particularly pathogenic for schizophrenic symptoms like hallucinations and delusions.
So it's not just the prefrontal cortex generally, but very specific parts of it and surrounding regions. Almost like finding the exact neighborhoods where problems are arising. Are researchers moving beyond the prefrontal cortex in newer studies, expanding their anatomical horizons?
Absolutely. While the PFC is crucial, newer studies are wisely expanding into additional critical brain regions to get a more complete picture of circuit-level dysfunction.
Like where?
The hippocampus and amygdala, for instance, are increasingly implicated. These are key for learning, memory, and emotion, and are central to disorders like MDD and PTSD.
Makes sense. What else?
Then there's the striatum, composed of the caudate nucleus and putamen. It's been identified as a central component of basal ganglia circuits.
And its function is disrupted in schizophrenia.
Its function and connectivity are disrupted, yes, making it a crucial area of study. It's also a focus of iPSC-derived ventral forebrain organoid, VFO, studies.
Those lab-grown models again, what are they showing in the striatum?
They're revealing that medium spiny neurons, MSNs, a primary cell type in the striatum, are often at an immature stage in these disease models, hinting at developmental issues.
And we mentioned the NTS earlier.
We did. The nucleus tractus solitarius, NTS, in the brainstem, critical for gut-brain and heart-brain connections, showing those specific inhibitory neuron changes in epilepsy combined with depression.
And what about other, perhaps less obvious brain areas that might be playing a hidden role?
There's growing interest in locus coeruleus norepinephrine, LCNA, neurons.
Tiny clusters in the brainstem.
Yes, producing norepinephrine, a neurotransmitter involved in stress responses, arousal, attention. They're studied for how their activity is modulated by various substances and their involvement in stress responses.
Like what substance?
For instance, prostaglandin E2, or PGE2, can have suppressive effects on these neurons via the EP3 receptor, influencing mood and anxiety.
Interesting. And even the sense of smell was relevant.
Intriguingly, yes. The olfactory neuroepithelium, the tissue lining the nasal cavity.
Where we smell things.
Right. It's being explored as a non-invasive model for schizophrenia. Findings suggest it contains a cell type comparable to those found in the developing embryonic brain, particularly mesenchymal cells.
Why is that useful?
Well, there's a high correlation between schizophrenia and anosmia, or loss of smell, which is sometimes seen in the disorder. So it's potentially a way to study brain-like cells without directly accessing the brain itself.
That's quite a wide anatomical net researchers are casting. Now let's talk about the cellular level itself. What kind of deep cellular profiling is happening? Which specific cell types are researchers isolating and analyzing?
They're delving into the intricacies of various brain cell types with unprecedented detail, moving far beyond just classifying them as neurons.
So starting with neurons.
Starting with neurons, we see particular focus on both excitatory and inhibitory types. Excitatory neurons are showing synaptic dysfunction and transcriptional changes in schizophrenia, indicating problems with transmitting signals.
And the inhibitory ones, the JBertic cells.
Inhibitory neurons, including GABAergic interneurons, are crucial for balancing cortical microcircuits and behaviors. For example, specific VIP and ID2 expressing interneurons are significantly affected by the 15q13.3 microdeletion.
Highlighting their vulnerability.
Exactly, in neurodevelopmental disorders. These inhibitory neurons are also identified in the NTS, with specific upregulation in temporal lobe epilepsy with depression, underscoring their role in comorbidity.
And the medium spiny neurons in the striatum.
We also discussed medium spiny neurons, MSNs, specifically D1 and D2 subtypes, found to be at an immature stage in iPSC-derived striatal organoids, highlighting developmental arrests in disease models.
Okay. Beyond neurons, what about the supporting cells, the glial cells? They seem to be popping up a lot.
Glial cells are indeed taking center stage, particularly astrocytes and microglia.
Astrocytes, the star-shaped ones.
That's them. They support neurons, regulate the blood-brain barrier, and they show pathology across schizophrenia, major depressive disorder, and autism spectrum disorder.
So they're not just passive support.
Definitely not. They also exhibit dysregulated genes related to neurogenesis and mitochondrial ATP synthesis in the context of 22q11.2 deletions. This indicates they're actively involved in disease processes.
And microglia. The immune cells.
Microglia are the brain's resident immune cells, constantly surveying the environment. They are deeply implicated in immune dysregulation and neuroinflammation across all three of these disorders.
With specific types identified.
Yes, with specific subpopulations identified, such as the interferon microglia in chronic stress models relevant to MDD. Single-cell studies prioritize microglia as schizophrenia-relevant cell types, especially in the cortical plate, due to significant correlations with disease status at the ATAC-seq level.
Meaning changes in their genetic accessibility.
Right, how open their DNA is.
Mm-hmm.
Then you have oligodendrocytes and oligodendrocyte progenitor cells, OPCs, also profiled in detail.
Their role is myelination, insulating the nerve fiber.
Exactly. Crucial for efficient signal transmission and overall brain connectivity.
So it's a holistic view, not just the neurons. Every cell type seems to be contributing to the overall picture of health and disease. What about the very early developmental cells, the building blocks?
That's a critical area, particularly for understanding neurodevelopmental disorders. Neural stem cells, NSCs, and neuroprogenitor cells, NPCs, are key to understanding neurodevelopmental signatures.
The foundational cells.
Yes, they give rise to all the different cell types in the brain. Studies are identifying novel behaviors in these cells, like overactivation, as seen with quiescent neural stem cells in SHNK3 knockout models.
And problems with multiplication or specialization.
We're also seeing altered proliferation rates, they multiply too much or too little, and specification issues, where they fail to develop into the correct cell types. For instance, RFX3 deficiency alters neurogenesis, impacting the birth of new neurons. And
FEZ1.
And FEZ1 deletion leads to abnormal expansion and ectopic localization of HOPX-negative outer radial glia, ORG, which are crucial progenitor cells for cortical development.
Meaning cells end up in the wrong place.
Essentially, yes. This granular detail helps us pinpoint the earliest points of potential divergence from healthy brain development.
Are there other cell types becoming more relevant? Perhaps connecting the brain to other body systems, hinting at a more systemic view?
Yes, increasingly so. Endothelial cells and immunocytes are being recognized for their crucial involvement in blood-brain barrier integrity and overall immune infiltration into the brain.
Endothelial cells line the blood vessels.
Correct. And the blood-brain barrier acts as a gatekeeper. This is particularly relevant in conditions like TBI and schizophrenia, where endothelial cell transcriptional changes are observed, indicating a compromise in this barrier.
So it connects brain health to the wider immune and vascular systems.
Exactly. Their role highlights the intricate interconnectedness, moving beyond just the neurons and glia within the brain itself. Segment 3. What are the key transcriptomic findings?
Okay, this is where we get into the nitty-gritty, right? What's actually changing at the gene expression level? The specific instructions being turned on or off in individual cells. Let's start by diving deep into the transcriptomic landscape of schizophrenia. What are we seeing there through the single-cell lens?
In schizophrenia, the transcriptomic findings are quite striking, offering a really granular view of the disease. We see clear evidence of immune system dysregulation.
How so?
Well, there's a widespread suppression of immune gene programs in blood cells, which might seem counterintuitive for a condition with inflammatory components.
Right, you'd expect maybe overactivation.
But conversely, in brain tissue, there's increased infiltration of immune-related cells, particularly microglial cells. And these show a significant correlation with schizophrenia-associated genes.
So suppressed in blood, maybe more active or infiltrating in the brain.
It seems like a complex interplay. Specific genes like S100A2, CCL14, and HLA-DRA are recurrently dysregulated, signaling changes in immune cell function and communication.
Even endothelial cells are involved.
Even endothelial cells in the prefrontal cortex show specific transcriptional changes related to the vascular system, like PCAM1, semaphore, and VESF1. It supports a broader inflammatory or vascular involvement beyond just the neurons themselves.
So a complex immune picture. What about the brain's energy factories? Are mitochondria involved in schizophrenia?
They absolutely are. We see significant mitochondrial and metabolic alterations. For example, the 3q29 dilution, which is strongly associated with schizophrenia, leads to a widespread decrease in the expression of genes related to mitochondrial energy production.
So less energy being made.
Potentially, yes. There's even evidence of shifts in the stoichiometry of respiratory chain complex proteins, meaning the balance of protein components that make up the energy production machinery is off.
The balance is wrong.
Right. This indicates a fundamental disruption in how brain cells generate energy, impacting overall cellular function. Additionally, we see alterations in the EIF2 signaling pathway, which is involved in protein synthesis and stress response, further hinting at cellular stress.
And what about the core function of neurons' communication? Are there transcriptomic signatures of synaptic problems?
Yes, definitely. We see widespread synaptic and neurotransmitter system dysfunction. In excitatory neurons, gene expression is enriched in terms related to sodium ion export, regulation of calcium ion-dependent exocytosis, synaptic vesicle transport, and neurotransmission.
So the whole machinery for sending signals is affected.
It appears so.
Yeah.
The intricate machinery responsible for releasing neurotransmitters and sending signals across the synapse seems impaired. For example, SET1A insufficiency directly suggests dysfunction of synaptic vesicle exocytosis, as we discussed earlier.
And this links back to the genetic risk factors.
It does. Key schizophrenia-associated genes identified in human GWAS studies, such as TOC2, SCAF1, and NESON6, are significantly enriched in the differentially expressed genes of excitatory neurons, highlighting direct links.
What other communication systems are affected?
We're also seeing issues with snare complex genes, critical for vesicle fusion and neurotransmitter release, and disruptions in endocannabinoid signaling, which modulates synaptic activity. It paints a picture of widespread communication breakdown.
That makes perfect sense, given how central communication is. Does single-cell RNA sequencing also tell us something about the developmental origins of schizophrenia, maybe even before symptoms appear?
It does, profoundly so. There's a strong neurodevelopmental signature. Genes identified as schizophrenia risks often show significantly different expression patterns between prenatal and postnatal tissues.
Suggesting they're important early on.
Suggesting their involvement in early neurodevelopment, yes. For instance, EEFSC, a novel risk gene, is associated with gestational duration, indicating a role even before birth.
Wow. And in the organoid models.
Furthermore, aberrant neural progenitor cell proliferation and differentiation, meaning these early building blocks don't multiply or specialize correctly, are observed in human organoid models of the 22q11.2 deletion syndrome. This is consistent with impaired neuronal maturation and neurate complexity.
So it supports the idea that schizophrenia starts very early in brain development.
It strongly supports the long-standing neurodevelopmental hypothesis of schizophrenia, pinpointing very early events in brain formation as crucial vulnerability points.
Okay, let's shift to major depressive disorder and chronic stress. What transcriptomic insights are emerging there, and how do glial cells factor in?
Here, glial cells are central, particularly astrocytes and microglia. They show disruptions in metabolic genes and immune pathways.
Microglia again. What's happening with them in stress?
In chronic stress models, microglia exhibit that robust downregulation of interferon-related genes, IRGs, especially in that distinct subpopulation, the interferon microglia.
And this involves the ATF3 factor?
That downregulation is speculated to be an IFN-independent mechanism, potentially involving the transcription factor ATF3, which acts as a negative regulator.
Are other processes disrupted in MDD models?
Beyond that, we're seeing significant disruptions in glycosylation.
Adding sugar molecules to proteins.
Yes, crucial for cell recognition and signaling. We also see impacts on the gut-brain axis, the communication system between the digestive system and the brain, and metabolic genes like FARS2 and SLC25A21 are showing altered expression, indicating fundamental changes in cellular energy and function.
So a targeted dampening of immune response in specific microglia, plus broader metabolic and even gut-brain connections. What about mitochondrial changes in depression, given their importance in schizophrenia?
We absolutely see mitochondrial stress as a recurring theme here, too. Mitochondrial biogenesis pathways, the processes for making new mitochondria, are impacted, especially in GABAergic neurons, which seem linked to stress vulnerability.
Any specific genes linking mitochondria and MDD?
In terms of specific gene insights, HTD2, an enzyme crucial for mitochondrial fatty acid synthesis, is found altered in mice with IRSP53 deletion. This is significant because HTD2 has been linked to neurodegeneration and brain function changes relevant to MDD. It points to a direct link between cellular energy production and mood disorders.
Now, let's explore the transcriptomic unfoldings in autism spectrum disorder. What's the single-cell perspective revealing here, especially regarding early brain development?
In ASD, a major focus is on neural stem cell, NSC, and neural progenitor cell, NPC, abnormalities.
The early cells again.
Exactly. For example, the SHANK3 knockout model shows that overactivation of quiescent neural stem cells, those dormant reserves we talked about.
Is there potential for intervention there too?
Excitingly, yes. Epigenetic modulation of these cells is being explored as a mechanism to potentially rescue synaptic dysfunction and behavioral abnormalities.
What about the 22q11.2 deletion models in ASD context?
In the 22q11.2 deletion model, patient-derived organoids reveal lower proliferation rates and slower differentiation of patient NPCs into neurons, along with reduced neurate complexity, meaning fewer intricate connections form. So clear developmental delays and impaired neuronal maturation.
Any other interesting gene findings in ASD, perhaps those impacting how brain cells organize themselves?
Yes. Human cerebral organoids with a FEZ1 deletion show an abnormal expansion of HOPX negative outer radial glia and a reduction of HOPX positive ORG.
These guide brain development.
Yes, they're critical cells that guide the development and organization of the cerebral cortex. This deletion leads to ectopic localization of neuroprogenitors, early brain cells ending up in the wrong place, increased cell mobility, and defects in cortical layer formation.
So problems with the brain's fundamental architecture.
It suggests a crucial role for FEZ1 in modulating neuronal progenitor subpopulation specification and migration. How the brain's architecture is laid out. Similarly, RFX3 deficiency leads to altered neurogenesis and significant downregulation of genes related to neurodevelopment and synaptic function, including those involved in synapse assembly and organization.
And what about the immune system in ASD at this granular level, building on what we heard earlier?
The IL-6-HIF1A immune axis is activated, particularly in microglia. Single-cell sequencing reveals increased expression of JUN-HI1A and potential diagnostic markers like CDKN1A, LYN, and SLC16A3 in reactive microglia MG2 and ASD.
So neuroinflammation seems key.
It solidifies the role of neuroinflammation in the pathogenesis, pointing to these specific immune pathways as contributors. Beyond direct gene mutations, epigenetic dysregulation like DNA methylation and histone modifications is also actively studied for its role in ASD pathology.
Highlighting how gene expression can be altered without DNA changes.
Exactly, often influenced by environmental factors interacting with the genetic blueprint. Well, segment 4. What's shared across disorders?
Okay, we've covered a lot of ground looking at individual disorders through the single-cell lens. Now here's where it gets really interesting and frankly quite thought-provoking. When we step back from the specifics, what common patterns are emerging from these single-cell studies? Are there shared underlying mechanisms across these seemingly distinct neuropsychiatric conditions, hinting at common vulnerabilities?
That's a crucial question, and the answer is a resounding yes. Across these conditions, which often manifest so differently, several recurrent biological patterns are emerging from single-cell analyses.
So it's not just isolated problems.
No, these patterns suggest common underlying mechanisms, even if their specific manifestation or the primary cell types involved might differ. There appear to be shared, fundamental vulnerabilities in how the brain develops, functions, and responds to stress.
So what's at the top of that list of shared patterns? What's the most consistent finding you're seeing across these diverse conditions?
Synaptic dysfunction is a pervasive theme, consistently appearing at the forefront. This is especially noted in excitatory neurons.
The brain's main communicator.
Responsible for transmitting signals, yes. In schizophrenia, studies highlight issues like altered synaptic vesicle exocytosis and neurotransmission, contributing to impaired brain function. It's like the fundamental talk between neurons is garbled.
And in ASD.
In autism spectrum disorder, problems with synaptic maturation and the expression of key synaptic regulators are observed, impacting neural circuit development, how the brain wires itself. For instance, RFX3 deficiency directly impacts synapse assembly and organization.
And in epilepsy with depression.
And in epilepsy with co-occurring depression, the enrichment of GABAergic synapse pathways suggests a widespread disruption of the excitatory-inhibitory balance, which is fundamental to healthy brain activity.
So how neurons communicate and the very structure of those communication points is a common thread of vulnerability. What else are we seeing consistently?
Mitochondrial stress and energy metabolism shifts are consistently observed.
The cell's power plant's sputtering.
Pretty much, yeah. Evidence of mitochondrial dysfunction is found in schizophrenia, particularly linked to the 3q29 deletion, affecting genes involved in energy production and respiratory chain complexes.
And in MDD.
In major depressive disorder, mitochondrial biogenesis pathways are impacted, especially in specific neuronal populations like GABAergic neurons under chronic stress. Genes like HTD2, that mitochondrial fatty acid synthesis enzyme, are implicated.
And epilepsy.
And in epilepsy-related depression, mitochondrial-related genes are significantly elevated in glial cells. It truly seems the brain's energy production system is a common point of fragility.
That's a fascinating link, energy disruption, playing a role in such diverse conditions. And what about the non-neuronal cells, the glial cells? Are they involved across the board as well?
Absolutely. Glial activation and neuroinflammation are consistent findings. Glial cells, including astrocytes and microglia, consistently take center stage in the pathology across multiple disorders.
Astrocytes are involved everywhere.
Astrocyte pathology is observed across SCZ, MDD, and ASD. We saw those dysregulated genes in 22q11.2 organoid astrocytes involved in neurogenesis and mitochondrial ATP synthesis. They're not just passive bystanders.
And microglia's role?
Microglial involvement is particularly highlighted in ASD through the IL-6HIF1A pathway. In chronic stress models for MDD, microglia exhibit that immune gene downregulation and morphological changes. In schizophrenia, microglial cells show a strong correlation with disease status. And TBI can induce neuroinflammation with immune cell infiltration.
So the brain's immune response system is a core shared vulnerability.
It certainly appears to be.
And finally, what about the very early stages of life? Are these disorders originating from fundamental issues in brain development?
This is perhaps one of the most profound commonalities. A strong neurodevelopmental signature.
Suggesting early disturbances matter later on.
It suggests that early developmental disturbances contribute significantly to later symptoms and disease emergence, yes. In schizophrenia, those prenatal versus postnatal gene expression differences underscore developmental origins. Aberrant neural progenitor cell proliferation and differentiation are seen in models.
And in ASD.
In ASD, abnormalities in neural stem cell activation, altered neurogenesis, impaired differentiation trajectories, disrupted neural migration patterns like with FEZ1 or RFX3 deficiencies are prominent features. The impact on interneuron development from deletions like 15q13.3 further supports this early vulnerability.
So many of these disorders may have roots in how the brain initially forms and wires itself.
It strongly suggests that, yes, long before symptoms become apparent.
It's truly remarkable how these very specific cell-by-cell studies reveal these broad, common themes, almost like finding recurring motifs in a complex symphony. But while these commonalities exist, I assume each disorder still maintains unique profiles. It can't all be exactly the same.
That's a critical point, and you're absolutely right. While the shared patterns provide invaluable insights into overarching vulnerabilities, each disorder does indeed retain unique profiles that highlight distinct pathologies.
Different pieces of the puzzle.
Exactly. Some shared, some unique. For example, in MDD, we see that specific immune glial bias with the distinct microglial downregulation of interferon-related genes that's not as prominent elsewhere.
In schizophrenia.
Conversely, schizophrenia often shows a particular interneuron vulnerability with specific impacts on interneuron development from genetic factors like the 15q13.3 microdeletion. That affects precursor cell proliferation and leads to behavioral issues.
So it's a tapestry, shared threads, unique patterns.
Precisely. Understanding both the commonalities and the specificities is absolutely key to developing effective targeted treatments down the line. Segment 5. Who's driving this research?
This incredible work, revealing patterns gene by gene and cell by cell, doesn't happen in a vacuum, does it? It requires massive collaboration, cutting-edge technology, and huge data infrastructure. So who's driving this research? And what are the essential tools and resources they're using to make these discoveries?
You're absolutely right. The sheer scale and complexity mean that large-scale collaborations and centralized data resources are fundamental. Think of it like a global scientific village working together.
Which groups are key?
Consortia like PsychENCODE, the Psychiatric Genomics Consortium, NEGC, and the Genotype Tissue Expression GTX project have been absolutely critical. Without them, we wouldn't be nearly as far along.
What have these major consortia provided that's so essential? What's their unique contribution?
They provide several crucial elements. Firstly, harmonized single-cell data sets. This means data from many different labs are processed consistently, allowing researchers worldwide to combine and compare analyses.
Standardization is key.
Hugely important. Without it, integrating findings would be far more difficult. Secondly, they offer brain-wide EQTL maps.
EQTLs, linking genetic variants to gene expression levels.
Exactly. So these maps link specific genetic variations to gene expression in different brain regions and cell types. This is key to understanding how genetic predispositions translate into altered gene activity in specific cells.
And they provide tools, too.
And thirdly, yes, they develop sophisticated tools for data integration. This allows researchers to combine genetics with cell type-specific expression profiles, so you can explore not just which genes are implicated, but where, when, and in what specific cell types these influences are acting, moving beyond broad strokes to really granular detail.
That's a huge undertaking, enabling a more global and integrated view. Are there other significant data resources contributing?
Yes, the Common Mind Consortium, CMC, and BrainCloud data provide extensive RNA-seq data from postmortem human brain tissues, including the dorsolateral prefrontal cortex, DLPFC, for schizophrenia and control subjects.
Real human brain tissue data.
Invaluable for detailed gene expression studies across developmental stages, offering crucial insights into how genetic risk manifests over time. And for creating cellular models, the Human-Induced Pluripotent Stem Cells Initiative, HIPSI, provides individual-level genotype data for iPSC lines.
iPSCs, the reprogrammed adult cells.
That's right. They're a game changer because they can be turned into any cell type, including brain cells. HIPSI provides the genetic info for these lines, crucial for genetic modeling in cell culture systems. It's almost like having a patient's brain cells in a dish.
So a global network of data and samples, bridging human tissue with lab-grown models. What about the actual advanced technologies powering these discoveries? What are the key innovations?
The cornerstone technology, of course, is single-cell RNA sequencing, scRNA-seq, itself. It offers that unparalleled resolution to capture cellular heterogeneity, the differences between cells and cell-type-specific gene expression patterns.
Seeing the individual cells.
Yes. Building on this, researchers are employing multi-omics integration, combining scRNA-seq with other data like ATAC-seq.
Measuring which DNA is open and active.
Right. And DNA methylation profiles and traditional GWAS data. This fusion allows for deeper, multidimensional insights, like getting a 360-degree view of a cell's biology.
Are there specific methods for this integration?
Yes, for instance, single-cell disease relevance score, CRS methods, are developed specifically to integrate cell-level information from scRNA-seq with polygenic signals from these multi-omics levels. Techniques like ketene are also used for epigenomic profiling, analyzing transcription factor binding sites, telling us how genes are regulated.
That's incredibly powerful, looking at multiple layers at once. What about the models they're using to study these diseases in action, in a dish or in animals?
The development of sophisticated modeling systems has been revolutionary. iPSC-derived brain organoids are providing human-specific preclinical models of brain development.
Different types for different brain regions?
Yes, forebrain organoids, ventral forebrain organoids, VFOs for striatal development, cerebral organoids for cortical development. These miniature tissues allow study of genetic deletions like 22q11.2, CHD8, FEZ1 in a human context. That's a significant step.
In gene editing.
Complementing this, CRISPR-Cas9 gene editing is used to generate specific genetic modifications in iPSC lines and animal models, like introducing SED1A mutations or 3q29 deletion lines to precisely study gene function and causality.
And animal models are still important.
And of course, animal models, primarily mice, continue to be essential for in vivo functional validation, behavioral testing, and studying gene-environment interactions. Think of the chronic stress models, TBI models, Shank3KOs. They bridge the gap between cellular insights and whole organism behavior.
It's a powerful combination. What other techniques are crucial for validating these findings, making sure genetic changes translate to functional changes?
Beyond gene expression, advanced imaging and electrophysiology are vital. Calcium imaging is used for in vivo visualization of neuronal activity.
Watching brain activity in a dish.
Literally, yes. Assessing how neurons are firing in organoids. High-density microelectrode arrays, MEAs, assess neuronal activity and network formation in organoid cultures, giving insights into communication.
And standard lab techniques.
Standard techniques like immunofluorescence and Western blot validate gene and protein expression changes, confirming the link. While Golgi staining provides detailed morphological analysis of neurons, looking at dendritic spines, the communication branches, these methods ensure we're looking at function, not just gene lists.
Finally, what about the digital side, the computational tools? Making sense of all this data must be a huge challenge.
They're absolutely indispensable. Imagine millions of individual cell readouts without powerful computers and algorithms. Tools like RNA velocity and pseudotime analysis using software like Svelo and CellRank infer developmental dynamics and differentiation trajectories.
Seeing how cells change over time.
Understanding the dynamic progression, not just static snapshots. Gene ontology, GO, and pathway analysis, IPA, interpret functional consequences, grouping genes into biological functions.
And understanding interactions.
Co-expression network analysis, WGCNA, and cell-cell interaction analysis, CellChat, identify genetic ensembles and help understand intercellular communication networks affected by disease. Seeing how cells talk or don't talk to each other. Tools like CyberSort and AWCell analyze immune cell infiltration.
All underpinned by statistics.
All underpinned by sophisticated statistical models, like linear mixed effects models and polygenic risk score calculations, ensuring rigor and reliability. These computational tools are the backbone that allows us to decode this vast information and transform raw data into biological insights. Segment 6, where do we go next?
So we've covered the disorders, the regions and cells, the transcriptomic findings, and the incredible technologies and collaborations. It's clear we're at a pivotal moment. Now, let's turn our gaze forward. As the field progresses so rapidly, what are the crucial areas for future research to truly translate these findings into actionable insights for patients? What's still missing? What should come next?
That's truly the million-dollar question, and there are several key areas researchers are prioritizing. First, while we've identified many differentially expressed genes and pathways, there's a critical, paramount need for deeper functional validation.
Moving from correlation to causation.
Exactly. Identifying an altered gene is just step one. We need to rigorously understand its causal role. Does it cause the disease, or is it merely a consequence?
How do researchers confirm that causality?
This requires rigorous studies using advanced techniques, CRISPR gene editing, iPSC models, in vivo animal studies. It's about experimentally manipulating these genes and seeing if the disease phenotype is recreated or rescued.
Which cell types are top priority for this validation?
Certain cell types consistently top the list due to their recurring roles across disorders. Astrocytes, GBA-ergic neurons, and microglia. For instance, understanding how epigenetic modulation of quiescent neural stem cells in ASD models directly impacts synaptic function is a prime example, showing how tweaking a gene has a tangible effect.
So functional validation is paramount. What's the second major area for future focus, especially given that recurring theme of energy disruption?
Given that consistent observation, targeting mitochondrial dysfunction and metabolism should be a major priority. These pathways need study not just as observations, but as active targets for therapeutic interventions.
Finding the mechanistic links.
We need to investigate the precise mechanistic links between the observed transcriptomic changes in mitochondrial genes and their functional consequences at the protein level, like in the 3q29 deletion research. The ultimate goal is exploring whether directly targeting mitochondrial function can ameliorate disease phenotypes.
A potential new avenue for treatment.
It could be, yes, something not previously obvious.
What else do we need to map out in the brain to get a more complete picture?
There is a major need to map understudied brain regions and populations. While the PFC is a hotspot, many crucial areas beyond it are underexplored at this level of detail.
Like which ones?
The amygdala, critical for fear and emotion. The ventral tegmental area, VTA, key for reward. The cerebellum, vital for motor control and increasingly recognized for cognitive emotional roles. Their detailed cellular landscapes in disease are less understood. The nucleus tractus solitarius, NTS, is also a target for epilepsy-related depression.
And it's not just about where in the brain, but whose brain, right? Diversity in research is key.
Precisely. Research must expand to include underrepresented populations, including females and individuals of non-European ancestries. This is crucial for generalizability and identifying potential sex or ancestry-specific disease mechanisms.
Ensuring treatments work for everyone.
Exactly. We know there are biological differences.
Yeah.
Overlooking them means treatments won't be as effective. The variability in iPSC donors highlights why this diversity is essential for equitable future treatments.
Finally, how can the field keep pushing the boundaries with technology? What's next to move beyond static snapshots?
The field must continue to embrace and integrate emerging technologies. Spatial transcriptomics is a key one.
Seeing gene expression in its anatomical context.
Exactly. Providing crucial spatial information about where cells are and how they interact in their natural environment, like seeing the whole symphony, not just individual musicians.
And combining more data types.
Further sophisticated multi-omics, integration genomic, transcriptomic, epigenomic, proteomic data at single-cell resolution will provide an even more comprehensive understanding.
What about AI?
And importantly, AI-based cell state modeling is rapidly evolving. Leveraging AI and machine learning like RNA velocity and cell rank allows reconstructing not just static snapshots, but the dynamic trajectories of disease progression at the cellular level,
Inferring how cells change over time in disease.
helping to infer differentiation pathways and cellular transitions in health and disease. This is where we move from observing to truly understanding the unfolding narrative of disease, allowing us to predict and potentially intervene. We're truly at a pivotal moment in psychiatric genomics. Thanks to single-cell technologies and these powerful analytical tools, we're no longer just guessing about the complex cellular and genetic underpinnings. We're truly tracing psychiatric disease gene by gene, cell by cell. It's a level of precision we could only dream of just a few years ago.
These deep dives into the cellular and molecular machinery of the brain are not just academic exercises, They are setting the stage for revolutionary changes in how we diagnose, understand, and ultimately treat conditions that have long baffled us.
Guiding us towards a future where our understanding is precise enough.
To truly make a difference for countless lives, it's incredibly hopeful.
Stay tuned for our next deep dive, where we'll explore how these precise, cell-by-cell findings are being used to discover novel biomarkers and, crucially, guide the development of truly targeted treatment strategies.
Until then, keep asking questions and keep zooming in on the incredible world within.